All right, so today we have sub-I presentations to start off, and then we'll have Dr. Scheurer giving a presentation in the second part of Grand Rounds. We're going to start with Brian Lien, who's a student from UC Irvine, who's now been with us for 3.5 weeks. It's wrapping up this week. He completed his undergraduate work at UCSD, where he did quite a bit of neuroscience research, including with in the lab of Dr. Huberman. He then moved on to UC Irvine, where he's been working hard towards a neurosurgery residency since the beginning of medical school. He has done some research also with Dr. Lim at University of California, San Francisco, and completed an ABTA fellowship in that lab during school. So he'll be presenting today on some of his glioblastoma research. Brian, looking forward to hearing what you have to say.
Great, thank you for that introduction. I'm going to share my screen. So hopefully everybody can see that. So the title of my talk is Long Non-Coding RNA Therapeutic Targets in Malignant Glioma. I'm a student at UC Irvine, but this work was done at UCSF in Daniel Lim's lab. He's a neurosurgeon scientist. I joined his lab before medical school and completed an ABTA fellowship during one of the summers during medical school. So to start off, why should you care about long non-coding RNAs? First of all, they're defined as RNAs at least 200 kilobases in length, and they don't code for proteins. This class of RNAs are actually very numerous in the human genome, and there has been an explosion of research on this class of RNAs in the past few decades, but the majority of these RNAs have not been thoroughly studied. And just to give you some perspective, there are about 20 to 25,000 protein coding genes, whereas there are estimated over 40,000 long non-coding RNA genes. And they were thought to be RNAs that were transcriptional noise, but it's what we found is that these genes play important roles inside the cell, and they play roles in cancer pathogenesis as well. They're thought to be attractive targets for cancer because they're highly cell type specific, more so than protein coding genes, and that is beneficial because theoretically there's less potential for off-target effects. So just a little bit of background on malignant gliomas. In adults, GBM is the most common, comprising 82% of cases. Standard of care involves surgery, radiation, chemotherapy. With respect to radiation, the toxicity of radiation really limits its dose, but it is quite effective. But of course, you all know there are side effects. Brainstem gliomas are a very common cause of malignant glioma in children, and diffuse intrinsic pontine glioma are the most common of the brainstem gliomas, comprising 80% of the cases. This disease is not one that can be surgically resected. Sometimes biopsies are warranted, especially if the child can be enrolled in a clinical trial using the biopsy results. And then radiation and chemotherapy are the mainstays of treatment. So despite our best efforts, the prognosis for malignant glioma is quite terrible, which is why research is really important in this area. And of note, any novel therapy that can complement the standard of care would be useful for the treatment of these diseases, but also for the design of clinical trials. So what we did is we used Crispr Interference. You all know Crispr as a gene editing tool. Crispr comes in different flavors. There's Crispr Interference, which involves a catalytically dead Cas9 protein, which doesn't have its nuclease function. It doesn't cut DNA, but it can block the transcription of any gene of interest. So what we did was called a pooled CRISPR screen. We found in RNA sequencing that in this particular cell line of GBM U87. There are over 5,000 link RNAs expressed. We did a pooled screen knocking down each of these long non-coding RNA genes. And we had a condition where there was no radiation, and we had a condition where the cells received fractionated radiation, 8 gray and 4 fractions. And the benefit of this technology is with you're infecting the cells with lentivirus, so the guide RNAs get incorporated into the genome, which is essentially a barcode, so that you can quantify the growth or loss of cells over time. So CRISPR has revolutionized scientific research in the life sciences. However, in the clinic, it's not been found to be therapeutically useful yet. We don't really have a way to get CRISPR inside of cells. For modifying gene expression, though, in gene therapy, antisense oligonucleotides are quite promising, in my opinion. They've already been FDA approved for SMA and DMD. So I'll backtrack a bit and show you that in our pooled screen, the most promising candidate sensitizing glioma cells to radiation was this gene called CTC338M, which was not very well studied. There was probably 1 or 2 papers that simply mentioned the gene. We renamed it LinkGRS1, and we decided to further evaluate this gene. When we knocked down this long non-coding RNA in GBM cells and DIPG, we had an effective knockdown, as you can see here. And you can see that the growth of GBM cells and DIPG cells decreased with knockdown of LinkGRS1. So an interesting thing about this gene LinkGRS1 is that it's primate conserved, so it's not expressed in lower mammals like rodents. It's primate specific, so any in vivo model involving rodents would be suboptimal. So we looked into using human brain organoids comprised of mature neurons and astrocytes to model the invasive growth of GBM and to test therapeutics. So you can see here we created the mature brain organoids of human cells. We seeded them with RFP, red fluorescent protein positive glioma cells. They attached to the organoids, and we found that they grew invasively over time. You can see here with the LinkGRS1 knockdown and with 0 gray radiation, there is still significant tumor burden over time. When we combine radiation with LinkGRS1 knockdown with the antisense drugs, you see that there's a significant decrease in tumor burden with 12 gray of radiation and 18 gray of radiation. And we quantified this and found the difference to be statistically significant. Another set of experiments involved CRISPR activation. So just like you can use CRISPR to inhibit transcription of genes, you can use CRISPR to increase the transcription of any gene of interest. So I wanted to look more into the function of this gene that was pretty poorly studied. So I engineered a U87 glioblastoma cell line to stably express DCAS9-DPR, which is the protein that, well, it has a domain that recruits transcriptional activators that increase transcription. Added the guide RNAs for LinkGRS1 into the cells to overexpress LinkGRS1. And there were 3 different guide RNAs that were designed. It seems like the third one, we were able to increase the relative expression of LinkGRS1, and you can see here that there was an increase in cell growth over a period of 2 days compared to the control that was statistically significant. And then lastly, wanted to look at if LinkGRS1 at the locus had any function. So I queried the gene enhancer database, which is an integrated database of human enhancer elements. So what I found was LinkGRS1, the gene locus, seemed to form looping interactions with a family of protein called TRIM proteins, and these have been implicated in various cancers. So it appears that the LinkGRS1 transcript has a function in cells and involved in cell proliferation, and the gene locus itself seems to have some enhancer function as well. So just to summarize, CRISPR interference screens can be used to identify therapeutic targets that sensitize malignant glioma to radiation. LinkGRS1 could be a potential gene therapy target to potentiate the effects of radiation in GBM and DIPG. And as mentioned, my working hypothesis is that this gene has dual function, with its gene locus having a function in regulating neighboring genes, and its transcript itself promoting cell proliferation in glioma. And then lastly, human brain organoids are a promising tool to model the infiltrative growth of malignant glioma and prioritize novel therapeutic targets. And then just wanted to acknowledge my PI, Daniel Lim at UCSF, and then all the lab members that contributed to this work. Happy to take any questions or comments at this time.
So Brian, I'm sure you know, there are tons of, and this is not intended to be discouraging, but there are tons of oncology studies that kind of dead end at this point at the organoid level or the like cell culture level. How would you try to translate this so that it doesn't just die here?
Yeah, it's tough because I agree with you, a lot of candidate therapies do fail at the cell culture organoid phase. I guess the advantage of the systematic CRISPR interference screen is you have multiple potential therapeutic targets. I think there's been some promising results with LinkGRS1. It's hard to say if it will actually lead to a therapy, but I think with the advantage of the CRISPR screens is we have a lot of different targets to look at.
I guess what I mean is what would your next step be if you wanted to try to take this further?
Yeah,
in the lab.
You could do xenograft models in rodents. You could test it in primates. That I think that would be probably the next step.
Does anyone else have any other questions?
Great work, Brian, with your CRISPR interference screens and these organoid models. Very interesting. And I guess kind of to go along Dr. Morgenstern's questions, I wonder perhaps if, this could be used to develop novel chemicals that might work within the pathway of GRS1. So I was wondering if you could delve a little bit more into exactly sort of the pathway that is thought to be affected and how GRS1 creates this radiosensitivity of GBM cells.
Yeah, I think the likely mechanism it's probably multifactorial. I think part of it is at the epigenetic level involving the enhancer elements. I think likely it's involving some cell cycle gene products as well. That would be my my thought on that.